Students, welcome to another section of Biochemical Pharmacology, BCH 407. Today, we shall take our lecture series 5, and the topic is receptors and distribution. The objective of this lecture will be 1, for you to be able to define what receptors are, then to name the types of receptor and comprehend the mode of action of receptors. Without wasting time, we would like to start by asking, what are receptors, or what is a receptor? They are specialized areas on the cell membrane, and they could be enzyme, transporters, or nucleic acid. Receptors are found on tissues and organs. Better still, we can say that receptors are found on the surfaces of tissues and organs. There are two basic types of receptors: intracellular and surface or cell surface receptors. Intracellular receptor or surface or cell surface receptors. When you hear cell surface receptor or surface receptor, is one and the same thing. So the two types are intracellular receptor, surface receptor, or cell surface receptor. They permit entry of drug molecules into target tissues or organs. Now let's look at intracellular receptors. The intracellular receptor is also known as internal receptor or cytoplasmic receptor. Any one of these can serve. You can see the structure there, the diagram there. The extracellular receptor is found in the cytoplasm, where they direct hydrophobic drugs. They allow the diffusion of hydrophobic drug across the cell membrane and interact with intracellular receptor in the cytoplasm. That is, the intracellular receptors are found within the cell membrane. The intracellular receptors are also known as the nuclear receptors. They are receptors for molecules like steroid hormones, thyroid hormones, retinoid, and vitamin D substance. Nuclear hormone receptors are a large family of transcription factors that bind directly to DNA to regulate the expression of target genes. They also regulate cellular response to hormones such as sex steroids, vitamin D3, adrenal steroids, and other metabolic ligands, and are involved in metabolism as well as development and reproduction. So you can see the importance of intracellular receptors. We must not forget that intracellular receptors are based in the nucleus. Although The ligands of intracellular receptor differ in structure. They are though structurally similar and they have intrinsic transcriptional activity. Like I said earlier, they are found in the cytoplasm of the cell and respond to hydrophobic ligand signaling molecules that are able to travel across the cell, the plasma membrane or cell membrane. Once inside the cell, they bind to proteins that act as regulators to messenger RNA synthesis to mediate gene expression. When a ligand binds to the internal receptor, a conformational change exposes a DNA binding site on the protein. The ligand receptor complex moves into the nucleus, binds to specific regulatory regions of the chromosomal DNA, and promotes the initiation of transcription. Again, we can see the signaling molecule binding to intracellular receptor and the signal molecule receptor complex now transverses the nucleus and interacts with this nuclear DNA. So internal receptors can directly influence gene expression without having to pass the signal to other receptors or messengers. That is by way of mode of action of intracellular receptors. Now, the structural composition of nuclear receptor is displayed. They are made of the following regions: the transcription domain, the DNA binding domain, the ligand binding domain, and the inhibitory protein. The inhibitory protein is always present in the inactive molecule. Now, figure A here represents the inactive receptor, while figure B depicts the active or the activated receptor. In the activated receptor, a cofactor protein will come alive by the binding of ligand to the ligand binding domain. That cleaves the inhibitory protein and causes the molecule to have a compositional orientation. By activation protein interacting with hormone response elements. In the whole, we have a conformational change which affects the target gene and lead to expression of protein. Now, the surface receptor or cell surface receptor. You can see the diagram above has depicted the picture of cell surface receptor. It is a transmembrane receptor. You can see it is cutting across the cell membrane with the groove here you can see the groove there for the binding of ligand. Now, cell surface receptors function by transmitting a signal through cell membrane. The ligand does not directly enter the cell membrane. They are involved in most of the signaling in intracellular organisms. Cell surface receptors are generally of three categories or types. We have the ion channel linked receptors, G protein linked receptors, and enzyme linked receptors. We shall consider each one of these in turn, starting with the ion channel linked receptor. Now, this picture here or figure depicts the ion channel linked receptor. You can see to my left, you have the receptor here. Again, transversing the cell membrane or spanning through the cell membrane and is closed. Then to my right, you see it open. You see the signal molecule that is there, waiting to bind to the binding site, to its binding site. An ion channel linked receptor bind a ligand and open a channel through the cell membrane that allows specific ions such as sodium, calcium, magnesium, and hydrogen to pass through. To form a channel, cell surface receptor transverse the extensive plasma membrane in order to interact with the phospholipid tail that form the center of the plasma membrane. Now, the amino acid in the outside of the spanning protein, that is the receptor, are hydrophobic in nature, while those lining the inside of the channel are hydrophilic, so as to allow for the passage of water and ions. When a ligand binds to the extracellular region of the channel, there is a conformational change in the protein structure that allows ions to flow or to pass through into the interior of the cell. The G protein linked receptor, on the other hand, have a different mode of action. First, the G protein linked receptor binds a ligand and activates a membrane called G protein. G proteins are a heterometric molecule with three subunits, alpha, beta, and gamma. When a signaling molecule binds a G protein coupled receptor in the plasma membrane, a GDP molecule associated with the alpha subunit is exchanged for GTP. Now that action activates the G proteins. The activated G proteins then interact with either an ion channel or an enzyme in the membrane. The beta and gamma subunits dissociate from the alpha subunit, and a cellular response is triggered either by the alpha subunit or the dissociated beta pair. Hydrolysis of GTP to GDP terminates the signal. Now look at this structure very well, or the figure that is presented. Take a look at it very well. Now you see here that a signal molecule, this is free, where you have the heterotrimeric G protein is free of any signal molecule. Then you have the alpha, beta, and gamma subunit together attached to a GDP protein. Now, upon the receipt of signal molecule, the protein is activated, thereby allowing the GDP that is attached to alpha, beta, and gamma subunits to exchange for GTP. When that exchange occurs, alpha and beta subunits dissociate from the molecule, and then there will be some intracellular events that will eventually lead to response, or that will intracellularly lead to internal or cellular response. Now, when this protein is to be deactivated, it loses a pi molecule to form GTP. And then the alpha, the gamma beta subunit will now merge with the alpha subunit, and the protein becomes inactive. Now, enzyme-linked receptors are cell surface receptors with intracellular domain that are associated with an enzyme. The enzyme, this receptor normally have large extracellular and intracellular domains, but the membrane spanning region consists of a single alpha helical region of the peptide strand. That is by way of description. We shall see the structure later, or the figure later. When a ligand binds to the extracellular domain, a signal is transferred through the membrane and activates the enzyme, which set off a chain of events within the cell that eventually leads to a response. An example of enzyme-linked receptor is the tyrosine kinase receptor. The tyrosine kinase receptor, as shown in the diagram here, can be activated by phosphorylation of the kinase, and can be deactivated by dephosphorylation of the kinase. The tyrosine kinase receptor transfers phosphate group to tyrosine molecule. Signaling molecules bind to the extracellular domain of the two nearby tyrosine kinase receptors, which then dimerize Now, for a better understanding of enzyme-linked protein or receptor, you can see this structure here or this figure here. The plasma membrane, you can see the protein. So we refer to it as a transmembrane protein that has a transmembrane domain. We have the ligand binding domain as well as the active enzyme domain, because the ligand will bind at the extracellular surface of the cell, while activity will occur in the internal part or intracellular domain of the cell. Phosphates are then added to tyrosine residue on the intracellular domain of the receptors and can then transmit a signal to the next messenger within the cytoplasm. Now we have looked at the ion-linked, G-protein linked, and enzyme-linked receptors and their mode of action. We know that these three receptors I mentioned are part of the surface receptor, while the intracellular receptor is unique on its own. With that, we've come to the end of this lecture 5. It is simple enough. I told you from the beginning that it is easy for you to have an A. All you need to do is to read what you have been given and read your extra notes. You have a reference material that has been recommended for you. Just do yourself a little favor, spend time on it, and you are good to go. Now you can assess yourself with this question. 1. Define receptor and list the receptor types. 2. Explain ion coupled receptor and its mode of action. 3. Describe G-protein coupled reaction and its mode of action. 4. Enzyme coupled receptor is a surface receptor. Discuss. Thank you very much for your time, and we shall meet physically for questions and clearing of certain misunderstanding that you may have. Thank you very much once again. See you in lecture 6.